I'm Georgia and I'm a researcher with the Oceanomics team at the Minderoo Foundation, working closely with the Minderoo-UWA Deep Sea Research Centre. My role focuses on using environmental DNA technologies to expand our knowledge of deep sea environments. So environmental DNA, or eDNA, refers to any genetic material an organism releases into the environment. As this genetic material degrades rapidly in the environment once it's shed, we can use it to see which organisms were present at the time of sampling.
These are just plain filter papers that are going to be picking up the eDNA samples in the water column basically throughout as the land is going down.
The Deep Sea Research Centre recently completed an expedition to the Nova Canton Trough on board the Inkfish vessel RV Dagon. The Nova Canton Trough is a fracture zone south of the equator right near the island country of Kiribati. It's about 8,000 metres deep and is biologically unexplored. On board, researcher Ellen Thomas collected active and passive eDNA samples. The active samples were collected using Niskin bottles on the side of the deep sea landers. These Niskins close on the bottom of the ocean, bringing deep sea water up to the surface. The passive samples were filter papers and puffs attached to the side of the lander.
Yeah, so the guys just brought the surface sample Niskin in. I just released it into the bucket. We've got the 3 tubes coming up here to the pumps. Once it's done filtering, I'll take the housings into the hood because the hood is a contamination-free zone. And I'll unscrew the housings, take the filter paper out, cut them in half, and then put them in these small tubes.
And we're hoping that these passive samples will show us insights into the entire water column in this region. Now that the samples are back in the Oceanomics lab, we can extract the DNA, amplify the DNA of our target organisms, and sequence this material. For these samples, we're interested in everything that's down there, as very little is known about what lives in this environment. We'll be using a suite of eDNA assays to target a wide range of both vertebrate and invertebrate DNA. Hopefully, what we gain from these eDNA samples will complement and expand on what we've seen on the lander and submersible footage, providing us with a better idea of what inhabits this region.